we're lucky because Robert is willing to come here for a sabbatical whenever he can. And hopefully that will be next year. But whenever the borders are open, we may be actually be lucky enough to have Robert here working. And it would be a fantastic opportunity to start collaboration. So I encourage everyone to contact him now in the questions or simply to his email that I'm sure you can find very easily on the internet. And Robert, without further ado, whenever you're ready, are you in Australia now? Is that the Northern Territory?
Well, I was once in Australia. So thank you, Danny, for the introduction. That is really nice of you. And thank you for the invitation to the seminar, because that is also very prestigious. It is midnight, so it's already Wednesday here too. But you know, it's just amazing that with Zoom, and basically in a way, that's kind of some positives that we can talk about COVID. That, you know, we now invite speakers from all over the world, as Danny was our speaker a couple of weeks ago back in Montana. And I just am reflecting to his attitude to the talk. He actually had a really nice American t-shirt that he got from one of our common friend, Professor Dr. Ray Callaway, and just for be respectful, he was wearing that. And I thought, I don't have a t-shirt anymore from Australia, but I have probably a nice background, a picture that I took back in 1999 when I was there for a year. And I really had a good time as a university student. So today, I would like to talk to you about soil seed bank and BioCrust and restoration, as Danny already introduced that a little bit. And I'm trying to advance my slides. Okay. First of all, in my presentation today, I would like to talk about a story that resulted in this really sad looking landscape in our beautiful state of Montana. And I hope you agree with me that it could be also from Mars, almost other than we had some tree trunks out there, and it is in a way all man-made. But before I go really into that story, I would like to tell you where the area is, because you're so far from Montana. So the Google Earth image shows Montana, and that little tick mark out there, the yellow one, is exactly pointing out where the research area is located, which I will be talking about today. And I just counted it today. It is approximately 12,300 kilometers away from your place. So it's really far, almost couldn't be more further. All right, turning back to the base of the problem, we really need to understand what happened in this area. And for that, we would need to go back in time, approximately 100 years in history. The story started here in Butte, Montana, which this postcard is depicting, a very old postcard, actually, a scanned version of it. And it tells us that this is the richest hill in the world. And this is because here a very rich copper reserve was found in the late 1800s. And it was about the time when Edison discovered or created the light bulb. And that way, there was a high demand on finding something that has a great electrical conductivity, and copper happened to be one of that metals. And so the mining industry right away skyrocketed because of that. And that little place that you saw on the map literally electrified America. And I would say we are still paying the price for that. I just would like to double check. Can you guys just thumbs up whether you hear me? Okay, and see the share. All right, that is really good. So with that, you see that this is an early picture from Butte, Montana. You can see all the the smokestacks, and you can see the head frames, sorry, which are basically the places where people went down to the mines. You also see that the houses are around all these smokestacks. You see also rocks, waste rocks, and tailings around the houses. This is really a special thing in Butte because Butte is built on the mines, and that was because people just simply went to work from their home down to the mines, and they didn't know better, and they just accumulated all these contaminated mine waste around them. We should also know that smelting was going on, as the smokestacks are showing it, but there was a problem in the area. There was not really a big abundance of water. So people were looking for better places to find water where they can smelt the ore. And that place was found approximately 20 miles, approximately 30 kilometers away from Butte, Montana, in the Anaconda Pintler wilderness today. And there was a great abundance of water. So the smelting operations basically came over here. And by 1896, most of the smelting happened here in the Anaconda area. And the next picture shows how scary it was. You see the so-called smelter stack, which is the tallest masonry stack built in the world, and it's still standing. And for the history, by 1896, most of the smelting came over here, and the area processed, so these operations processed approximately 11,500 metric tons of ore per day. And the smelting stopped in 1981. So approximately for 100 years, there was milling and smelting in the area that produced high concentrations of metals and metalloids, for example, arsenic, lead, copper, cadmium, and zinc. And this polluted the air, the soil, groundwater, and surface waters as well. On top of that, as this following the next picture is showing, where this lady is standing, there's just a lot of wood, lots of timber piled up. And the area had really good coniferous forests, and they were helping to fire all these smelting operations, but the timber was also sold because it was a very good quality timber. So a century of smelting and deforestation resulted in this completely denuded landscape of approximately 777 square kilometers, which is a large, very large area. With the topsoils and organic layers completely gone, they were washed away. And today this area, so just for you to see, here's one of my students collecting data, and you can see this tree trunk and the approximate soil layer, the A horizon, could have been approximately here. And you see that at least 40 to 50 centimeter is missing. And that is, I would say, almost irreplaceable. Today, this area is part of the nation's largest Superfund sites. Here in the US, the Environmental Protection Agency has this Superfund program, which is responsible for the cleanup of some of the nation's most contaminated lands. And just going on, so this is actually today. These are my pictures, and the landscape doesn't really look nice. This is rhyolite, a volcanic parent material that is to be found in the area, and very sparse vegetation. Also, for you to see, we can suspect that there could be out here a lot of contamination from the smelting, but actually because of the rains and time and how the top soil layer washed away, these contamination levels really went down to I would say, a minimum level. So if you see arsenic in the soil samples we took was approximately 6 milligrams per kilogram, and copper 16, that's not much. We were talking before about thousands of milligram per kilogram of these metals and metalloids being present. But what also we notice is that the different kind of nutrients, the macronutrients, are very sparse, and also the pH is still low compared to what plants in general like. Because the area is part of the Superfund site, large-scale restoration action started in the area. There was a so-called top-to-bottom approach, which started to do these upland vegetation enhancements, so kind of on top of the hill. Then they were coming lower down, and they did gully best management practices, so managing gullies so that they shouldn't take that much water and sediments away, but kind of keep them in the area. And also, there was a kind of a riparian enhancement. You see here a helicopter bringing seeds and fertilizers up to the area. It's a huge area, as I told you. There's no roads that are going up. So you either walk up there or fly up there. And you see people doing things by air or by hand. These ladies here establishing these gully breaks to again stop erosion going off from the mountains. Okay. So now a question rises, a big question, which we can say, is there any resiliency in this land? In other words, would it be able to bounce back to its former state with or without restoration? Because that is a really important question before we start any kind of restoration topics, restoration research, or restoration projects. And as you see that there's already large-scale restoration projects going on, and I must say many times those projects, I'm really sorry, they start without really evaluating whether the methods that they're using work in. So for example, helicopter seeding, probably it's not a cheap approach to try to reseed a mountain. And if we don't know if it's working, if those seeds basically stuck or not, or will germinate, I don't know if we should really continue. So here's where our research came in, and our team, with our students, we wanted to test the following objectives, I would say research objectives. You probably now understand that the area has several limiting factors that could make life in a way impossible. So like if we question whether plant life at all is possible, we should test that before we do any kind of enhancements. And then the other thing is we could really evaluate resiliency of an area by testing the seed bank. You understand also now that the topsoil was washed away, which most likely would contain most of the seeds in the area, over 90–95%. So we are not really talking about a historic seed bank that was left there for hundreds of years, but kind of recent seed banks that are arriving to the area. It's still surrounded by forested, huge wilderness. And so we suspect that seed rains are coming, but whether they make it or they stay dormant or they try to germinate, we don't really know. So this little cartoon symbolizes our soil, and then theoretically there could be seed in there. Whether they're coming up or not, we should test. And very few studies so far really tested seed banks in regards of restoration, and especially if we're considering revegetation of contaminated areas, just like this study that I'm just citing here. So our approach was to this, that the seed bank that is theoretically out there needs a stimulus, needs a kind of an amendment, if we could do that somehow, that activates the dormant seed bank and makes the seeds germinate. And that way we can go for a better restoration outcome. And that was what we tried to do, but we really didn't need to think too far, because the ongoing restoration, large-scale restoration projects provided us materials for that. They were already using some kind of fertilizers and seed compositions that we wanted to test in a smaller scale and wanted to see whether they could really work and enhance the area. So we used the same amendments that were used out there, but not really being tested. We also wanted to see whether the amendments that are used out there are helping the biocrust development. And I don't know if we are using the same words for this, but under biocrust, we mean a soil surface community of organisms that could be compiled of mosses, lichens, different kind of algae, blue algae, for example, and other microorganisms that are part of this community, and they help the soil to aggregate and kind of keep in place as well. So if we could establish biocrust, that would be really helpful for the restoration of the area. So we wanted to see whether that could be achieved by these different amendments. And then there was another objective, because there was already a seed mix used in the area. We wanted to see whether the additional native seed augmentation would help diversity. So the approach for this was for sort of testing whether there is a possible existence of plant life in the area. We investigated the relationship between plant communities and different levels and types of contaminants. And how we did that, we did select 10 locations with 10 transects. And as you see it in this picture, we went from an unvegetated area and flipped the quadrates, the 1 by 1 meter quadrates, and wanted to see whether there is vegetation in there, and then also took soil samples in the same time, which we investigated by using an X-ray fluorescence analyzer, an XRF, and also measured the soil pH. And part of our samples were also investigated in the lab for testing the accuracy for the equipment. So here is a really interesting picture I took. This is one of our the earliest native plants in the area. It's called Douglasia montana, and it's a cushion plant. And I don't know if you see, my XRF is put up here, and I found levels of lead, for example, 5,311, I think it's parts per million. That's a lot of lead in the area. So, and I just took these samples just right next to this plant. So we found areas that had high amounts of concentrations of metals and low pH and so forth. And we wanted to see what really determines the existence of life out there. And right away, going into the results of this, this is a non-metric multidimensional scaling in which we are testing the abundances of basically our plots, the plants in there. And then we laid over environmental vectors that you see represented with these arrows. I'm sure most of you or all of you are familiar with this multivariative analysis. And this right away helped us to interpret what is really going out there. And we saw that if you see all of these plots kind of scattering around, and how the environmental factors, for example, pH, impacted what is out there, doing some statistics, we found that pH was one of the most important limiting factor for plant establishment. If the pH is higher, plants were happy and established. If it goes lower, they didn't do really well. I just point out this species down here, which was represented by the low pH plots. It's Agrostis cabra. It's a species, a native species that was never really found above pH 5. So this guy loved low pH. On the other hand, we also saw that higher pH and higher copper concentrations actually were associated with more invasive species. Just for Danny, to some kind of some fun facts about invaders. And so I don't really go any deeper into this. We could most of the time talk for, I don't know, 10 minutes about these kinds of graphs, but I don't think we have time for that. But the most important messages were told. The next things were, we wanted to see that the 10 most abundant species that we found out there, how did they lay out on these interaction plots when we investigated them against pH, for example. And in the case of native species that we found out there, they were quite tolerant, or several of them were quite tolerant with low pH. And I just point out that species here, Agrostis cabra, again, which again was an average pH of with an average pH of 4.2. Most of the plants, however, went closer to 6. We also, just again for Danny, we looked at exotic plant species, among them a lot of invaders, and one of Danny's favorite plants, Centaura staubi, spotted knapweed, was also to be found in the area. And you see that these exotic species were much more picky with pH levels, and they didn't really go lower than 5, like it's closer to the 6 and neutral, but nothing to be found in this area. We also tested these species against the different metals that could be of concern. Copper, which is also an important metal in a way that is important for plants, but it could also be too much, or lead in a way. You see the levels that we found. For example, in the case of lead, we were registering approximately 3,800 parts per million out there in some of the sites. And so we just put out first off against these two metals, the different species. And we saw that they, can kind of tolerate smaller, bigger levels. Definitely, if we think back of the soil samples that we took from an area of interest, these levels are much higher, and we could conclude that, yes, plant life would be possible because the levels out there were not as big, not as high regarding these metals. And we investigated the exotic species against copper and lead, and they were quite similar to the native ones, so it wasn't as striking as it was in the case of the pH. Arsenic and zinc were the other two with native species, again, the same native species, and again with the exotic species, just to kind of for you to see and have a feel for how much, for example, zinc can be out there in general in these areas. Okay, so we kind of have an understanding that plants tolerate pretty high levels of some of these metals. So I'm turning now to the second part of the research, where we wanted to test whether there is any seed bank in the area and whether there is any biocrust that we could activate. And for this, we conducted first of a greenhouse experiment for which we collected 120 gallons of soil samples from this area, which was the Joiner Gulch. It's a larger area, so we scattered our soil samples out quite nicely, I would say. This is Agustina, one of our students, and one of our professors, Catherine Zodro, who was part of also this project. And besides Pedro Marquez, they were co-authors on the paper we published not long ago on that. So we are collecting soils, and then we're bringing it back to the lab to test their seed bank, because that's how usually we test seed banks. We bring them back. It's really hard to recover really seeds and identify them that way. So we encourage them to germinate. We give them all kinds of good conditions, water, temperature, and in this case, we try to give them different nutrients. So you see the different kinds of treatments that we were using in this experiment. So there's two major levels, first off, for the treatments. Once we just wanted to see whether the seed bank itself with these different kinds of nutrient treatments will be coming up, will there be anything? And then on the other half of the trays, we added the seed mix that on these large scale experiments, the consulting firms were using. So that added up with 12 different kinds of treatments. And so here are the different fertilizers that we were adding: synthetic fertilizer and a biofertilizer product, with mycorrhizae. And I have all the details about these fertilizers, because you might be interested, well, what kind of fertilizers they were. It could be really important for future applications. Okay. As I mentioned, for the part where we tested the added native seeds, these were the seeds that we were adding to them. Nothing really crucial for us now to understand, but they were four species and 3 native grass species that we were adding, and these were just the calculations for the trays. All right. So, and here's how the experiment looked like. So here are these trays, these growing trays, that actually got approximately a gallon of soils, 120 of these trays in the greenhouse with the treatments. And Agustina Sabutei, one of our students that you saw before, she was checking on these trays daily. And in the beginning, she just needed to see whether, how the seeds, what's the dynamic, how the seeds are germinating and coming up. And then she reduced these times for just weekly or every second week to see changes in the trays. So you see that there's a lot of things growing. You don't know at this point whether these are the seeds that we were adding, or they are seeds that are voluntarily coming. And what I also would like to point out here is that bluish greenish layer, which in a way is biological crust. In a way, I would say this is a lot of blue and blue algae. And then later on, as the experiment proceeded, they were mosses coming in also into our trays. Okay, and they just sometimes, more or less took it over depending on the treatment. All right, so this figure shows the different species that we found in the soil seed bank experiment. And what I would like to point out, the volunteer species are marked with blue bars, and the seeded ones are with red bars. I would say there's not terribly many species that came out voluntarily, but one of them almost was present in all 120 trays that we didn't see it, and it was out there. It was Agrostis stolonifera. And then there were other ones, and some of these grasses, for example, are exotic, unfortunately. So the seeds present in the seed bank, they're not necessarily always just good, but there was something. So here's what we can see based on the species. And then I'm turning into the results where I show you the results of the treatments. So for us to understand these graphs, it is showing the effects of the different amendments. And as I pointed out, there's these two different groups. There's the control, where we only tested again what is originally in the soils. and we didn't add any seeds. And then mix, which is represented here, shows us where we added those 7 different species in our trays. And right away, there is a striking difference between the mix and the control. And quite obviously, when we add seeds, there will be more germinating, and that is right there. But if we turn back to maybe the best and original question, whether there is any kind of opportunity for us to activate seed banks with these amendments, one of the fertilizers, biofertilizer applied alone, significantly increased the number of forb seedlings in our treatments. And when we turn into where we added seeds, the effect was just different, completely the opposite. So for example, if we consider this biofertilizer, it's explained here, compared to the control and synthetic fertilizer, it behaved actually just worse. So depending on the combination and the treatment level, we could just play this out really badly, which can be seen according to this figure. Number of grass seedlings, that was the other group we were testing. Again, the striking differences were between the control and the mix group, where we added the seeds and where we didn't. No differences in the seeds emergence in the control group, but there were again just different kind of deviations when we added the seeds in there. And this is how the above ground biomass changed by applying the different amendments. And that could be again straightforward. Well, if we add fertilizer to plants and if we water them, most likely we will have larger biomass. And in a way, we see that quite well. We also had an interesting trend here. One of our fertilizer, the pink, contained mycorrhizae, and I believe that, yes, it has the E. So compared to control and to a couple of these other letters were actually for the control, definitely the mycorrhizae application brought bigger biomass. And that just came out later. So when we early on applied mycorrhizae in our fertilizer, it didn't change our seedling numbers, but it was in later stages when we harvested the experiment where we saw some differences with the mycorrhizae application in the biofertilizer. Okay, and here comes the next level, and this is very interesting to me, and I didn't want to bring in a lot of different slides about that because maybe after a while it just becomes too boring. So what we see here, we see of course all the different kinds of treatments and then the total biocrust cover in our trays. And right away we can see this is control and synthetic fertilizers, they were worse. But everywhere where we applied biofertilizer, those treatment groups significantly increased the biocrust number from like 30 something to almost 90. That's huge. And we actually made separate studies on how blue algae and mosses changed with this application. And all of them showed similar trends in a way as expected. And here comes another cool thing, that with growing biocrust cover, there is a positive relationship between growing biocrust cover and the above ground biomass of the plants, which could be a very interesting outcome. And it was observed in other studies that biocrust could help. In this case, in the case of Mendy Slade's study, they showed that the biocrusts inhibited the exotic plants, but they helped the native plants. We didn't check that phenomenon in our study, but we are really hoping that that could be true. And right now we are conducting the field, we are analyzing, I would say, the field part of this research. And I mean, I'm just really excited that we hopefully will find the same thing, that BioCrust will help the native plants to thrive. So just this trend, and then that the biological crust was activated, that largely by these amendments, to me, this is really good news. And for summarizing up this research, I just have the following conclusions. The existence of plant life, as you understood, was revealed. So it's totally possible for plants to take over these areas. And the area has a pre-existing seed bank that for some level could be activated with the amendments. Especially I'm kind of focusing on the forbs that were significantly successfully activated by the biofertilizer. And then one of the most exciting discovery here would be for me that amendments helped the BioCrust activation. And then also the BioCrust increased the vascular plant growth. But in the end, if we would like to reach a certain amount of diversity, we definitely would need to actually think about augmenting seeds as the large-scale restoration projects are doing it anyways. There are also some limitations to this study, as there are also always for greenhouse studies. We are creating these artificial conditions, of course, where we are watering and providing good temperatures, kind of optimal growing conditions for these trays. And that could be a little bit of a, whether the same thing would happen in the field, it's questionable. Also, the next slide shows us an area out there. This is Karen Nelson, the former director of the Society of Ecological Restoration. She was out in the field with us, checking out how exciting this whole thing is. As this is an area which was treated by the helicopters, and they seeded out they seeded out the seeds, and they put out fertilizers. And I'm just pointing it out. I'm not sure how well you guys could see that. There is this green crust that is very much visible. And that was almost always to be found in the areas that were treated with the biofertilizer out in the area too. And what you can also see, there's a lot of germination, at least here, of grass species, and there's a couple of forbs coming up. So a couple of take-home messages, just two of them, the most interesting things I would say. Dormant seed banks possibly could be activated by these nutrient amendments, or with some of them. And I would say for ecological restoration of such areas, it may be enough to just start succession by activating the cryptogamic crust, because afterwards, if we imagine that this site will receive seed rains from surrounding habitats that are native, why would we even care about bringing in seeds from a seed company, even if they're native? They're just new genetic material. So we probably would be just good with activating that BioCrust, which could help us to just receive the plants and make a bigger plant biomass in the end. And starting basically this succession could be the key to the successful restoration of these areas. And with that, I took up a good 40-45 minutes, I would say. And I would like to acknowledge to some of the people who worked with us, Krista Ivalic, who is our greenhouse manager, Zsuzsanna Szabó, who helped me doing the very hand-intensive labor, and of course Agustina, who I also thank, although she's also part of these papers, but I'm very thankful for her to be my master's students, and now she's a PhD at South Dakota State. Also, the Big Hole Watershed provided us funds and areas for conducting this research, and the Natural Resource Damage Program in Montana. They help us to also fund that cryptogamic research, which now is really focusing on this field part of the experiment. And I wish I could have actually showed you more graphs and more relationships in the field, measured relationships, but I feel really excited about the outcomes. And with that, I would like to thank you for your attention. I hope that was somewhat useful and you just kind of can see this nice Montana landscape with some problem areas, but providing those colorful spots, which could be, leading us to a brighter future. So thank you guys a lot for listening to me.